Welcome to my 7th lecture of genetic series. As I told you that I have tried to make our lectures and presentation in line of ICR syllabus and syllabus which are being commonly used in most of the universities of India. In this continuation, today we will be talking on quantitative genetics. One thing I have to share with you all with happiness that we have formulated one logo for our YouTube channel, which is perhaps evident before you. You might be able to see the same in my first slide, and this will have to be protected under IP in the Indian territorial condition. Then as I've told you that I will be talking with you about quantitative genetics today. Before starting the exact terminology of quantitative genetics, it is necessary to know something about terms which are correlated with the quantitative inheritance and various aspects which are related with the quantitative genetics. Very soon, as you all are aware that we most of the time use the terminology phenotype, genotype, qualitative trait, these all, so that it is very necessary to understand about what are phenotype. The phenotype, these are the phenotypic appearance. These are the appearance of a translated genotype. This is due to existence of genotype. Genotype have been translated in some phenotypic appearance. That may be in the form of morphology, in the form of chemicals, proteins, etc., etc. And these all appearance which might be due to this genetic architecture of an individual, this will be termed as phenotype. Number of examples are which the things which could be seen morphologically, shape, size, etc. These all will be the example for, of the phenotype. Like a person has blue eyes, red eyes, black eyes. This is the phenotypic trait in human being or in any other animal which will be carrying this type of characters. Then what are genotype? The set of the genes that create the phenotype. These may be the single gene in the case of monogenic characters, but in most of the cases these are the cassettes of gene. It means there will be involvement of more than one gene which is translating the same phenotype, a single phenotype. This is the condition that the phenotype will be same in and background of that phenotype the activity of various genes may involve. Then this gene cassette or the community of gene which is translating particular phenotype will be considered as genotype. Or for your ease, we have given the second thing, specific genes that express a certain phenotype. For example, capital W, capital W in the case of Drosophila, this genotype will be translating in the form of eye color. And then what are the qualitative traits? The contrary word of quantitative trait regarding which we will be talking today is qualitative traits. What are qualitative traits? Specific characters that are. What are the specific characters which will be defining the qualitative traits? Determined by monogenes. In very few cases, there may be involvement of oligogenes, but the strict definition of qualitative traits are which will be determined by monogenes or various alleles of a gene, multiple alleles of a gene. Okay, this will be considered as qualitative gene. In the case of the character which are being translated, being influenced or being inherited by qualitative traits, these will be very least affected by environmental conditions. It means there will be about negligible, you can say, as there no influence of environment regarding if you are talking about the qualitative traits. And these are also called as classical Mendelian traits. Most of the time before the if you go back in the science, then these will have to be termed as classical Mendelian traits, and these have distinct phenotypic classes and exhibit discontinuous variation. This is the point to be noted, that these exhibit discontinuous variation, whereas in the case of quantitative characters we will be talking about the continuous variation. There will be existence of some specific classes, and both all the classes will be translated, will be governed due to existence of the genotype in background, so that these will be considered as qualitative traits. Now I will have to come directly on quantitative traits. What quantitative traits? What are they? If we are talking about the definition only, then quantitative traits is specific characters that are determined by multiple genes. As I've told you that in the case of qualitative traits, these have to be influenced, these have to be inherited by monogenes or various alleles of a particular gene. And these influenced by environment factors, for example yield. These are variously influenced by the environmental condition, environmental factors, various environmental factors like daylight, existence of humidity, temperature. These all are various characters related, factors related to edaphology, which are related to the soil conditions. These will affect the quantitative traits. And the loci involved to influence the trait will be known as QTLs. QTLs will be very buzzword. These are being used in the case of molecular genetics or molecular inheritance regarding QTLs. QTLs are the loci. These are the loci which are related with the quantitative traits, various traits which are translating for the same phenotype. These are also called a metric traits. Actually, in the case of qualitative traits, these are classical Mendelian traits, whereas in the case of quantitative traits, these are also called as metric traits. In various thesis work, the researchers used to write down the quantitative or metric traits. They produce continuous variation. I told you in the case of qualitative traits, there is existence of discontinuous variation, but here is the existence of continuous variation. They produce continuous variation. These are widely influenced by environmental conditions. I told you that various environmental factors are very much of significance over here in the case of quantitative traits. Each gene has such a minor effect that they could be measured by various statistical methods only at the place of Mendelian method, whereas in the case of quantitative traits, they can be easily measured by counts and other Mendelian ratios, where in the case of quantitative traits, they will not follow exactly Mendelian trait if you are studying as a whole, so that the population study is the major thing, and various statistics which are related with the population study will be of evident significance regarding studying the quantitative traits. Now, what are the major characteristics of quantitative genes? Till now we were having the discussion about the quantitative traits, but now we will be having some specific points related to characteristics of quantitative genes. At the place of trait, we will be discussing about the genes. Single gene or allele have very minute effect of phenotype. As I have told you that various or multiple genes are multiple genes, not multiple alleles. In the case of qualitative inheritance, there will be involvement of multiple alleles, and there is the involvement of multiple genes. Then a single gene or allele have very minute effect on phenotype. On a phenotype, they will be having the minute effect, whereas the phenotype is due to the cumulative effect of all the genes which are involved for the translation of particular trait. And second one, individual locus among the complete gene cassette for a trait segregate Mendelian method. If you are talking about the single gene, then this single gene will follow. If the single gene means this will be follow the monohybrid condition of the Mendelism. It means law of segregation. They will segregate as per the Mendelian method, Mendelian law of segregation. And crossing over occurs between the genes. I have told you that these are the various genes, multiple genes. And if there is existence of two genes, it means there must be involvement of crossing over. So that in the case of quantitative genes, they will follow and they will involve the crossing over, and they separate in the coming generations, coming offsprings. Polygenes has pleiotropic effect. What are pleiotropic effect? The influence of a particular gene on various traits. Actually, as today we were talking about the quantitative genes, it means there is involvement of a multiple genes for a particular trait. And on contrary, there must be a chance, a condition where a gene affect more than two traits, more than one trait. And this condition also follow in the case of polygenes. Then deleterious to changing various factors of environment. I have told you that environment is the key factor. Environmental factors are of significant value in the case of inheritance of quantitative traits. Therefore, the quantitative genes or the minor genes will be deleterious to various environmental conditions. Now, quantitative inheritance is also called blending inheritance. There may be a question in the various examination that what is the synonymous term for this quantitative traits or quantitative genes, quantitative inheritance. Then regarding the this blending inheritance is this synonymous of quantitative gene. The example which have been taken in my previous slide, we have just reversed it, that in the case of one polygene pair or two polygene pair or three polygene pair, in all these cases the parental type is male and female parent or type are present on each and every terminals, in previous terminal of x axis and in last terminal of the x axis. It means the parental type is always two. One is of female type, one is of male type. Whereas for other combination, this combination is increasing, being increased. In the case of one polygene, one combination is there. In the case of two polygene pair, three combinations are there. In the case of this three polygene pair, five combinations are there. It means combination will be increasing. It means the number of parental type remains always same in the case of quantitative inheritance, whereas intermediate types will be increasing as per the involvement of more and more number of genes responsible for the translation of the trait. What is the possible origin of the polygenes? What are the possible origin points from which this polygene may be arises? First one is duplication of the chromosome part. Particular part or segment of chromosome may be duplicated. I will be taking one separate lecture on chromosomal aberration. Then I will make very clear about this phenomena. Then what type of inversion, duplication, these all sorts of blah 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 will be take place in the case of conversion of monogenes in the form of polygenes. Second one is polyploidy, or increase in chromosome number. That may be in the nature due to spontaneous different evolutionary forces, or this may be due to treatment with colchicine treatment. This may be there may be existence of polyploidy, and which will create the existence of polygenic traits from monogenic traits. Then what are the now the third major point by which these polygenes may be arises are mutation producing genes having similar effect. And now we have kept over here the clear to make more clarity about quantitative traits and qualitative traits. In the case of first one, these traits are related with the Mendelian traits, which are in most of the conditions governed by monogenes or oligogenes, which has been studied in the P. And the second one, Carl Reuter, as I have told you that Carl Reuter has studied very previously about the occurrence of the quantitative genes or quantitative inheritance. If you see very clearly, then in the case of Mendelian inheritance in F1, the dominance, it means the existence of the character. If you study this on the basis of value, the mathematically or statistical study, it will very much clear that all the observation, all the individual particular observation will favor the dominant gene. Whereas in the case of this quantitative inheritance, they will follow as intermediate product after crossing the parents. In the F1, in the case of Mendelism, they will follow the dominant pattern. It means they will aggregate at dominant site regarding that particular trait. And in the case of this quantitative traits, they will lie on the intermediate, in between of the both parents, in between of the both parents. And in F2, you may see regarding Mendelism that there are clear-cut two peaks are there. One peak will follow the recessive traits, and second will follow the dominant traits. It means there is very clear-cut segregation pattern among recessive and dominant in the case of Mendelian methods, Mendelian these ratios, whereas in the case of this study which has been taken over by Carl Reuter, these will follow the normal distribution. What is the normal distribution? Perhaps I will take in the coming slide. In the case of this quantitative inheritance, between the recessive and dominant traits, the intermediate, the recessive and dominant, the single pattern, the single individual will follow the pattern of recessive and dominant traits, whereas the most of the population will follow the intermediate pattern. And there is a curve, clear as normal distribution, normal curve, in which there is one peak, and some individual lie before peak and some individual fall after peak, but between the limit of recessive and dominant, this value of the particular trait. Now we have taken we would like to give some examples related with quantitative inheritance. As I've told you that the study which has been taken over by this kernel color of wheat by Carl Reuter, we have given a one separate lecture with the name of multiple factor hypothesis, in which 1:4:6:4:1 will be the evident due to involvement of more than one genes for translation of the similar trait, the same trait. And so that I am not going to elaborate over here again. You may watch my previous lecture, which is totally dedicated on this multiple factor hypothesis. In the case of skin color in human, this is very important here. Therefore, we would like to give some glimpses, some detail about skin color in human, which has been studied by Davenport in 1913. The human skin color is deciphered by involvement of two pairs of genes. If we consider one pair of gene in homozygous form, capital A, and second one is which capital B, then capital A, capital B, this gene will result the negro skin color in the case of human. Negro skin color बिल्कुल काला होगा, just like black. Then अफ्रीकन, जिसे अफ्रीकन रेस कहा जाए आप, तो ये जो निग्रो रेस होती है, this skin color, this black color of skin is due to involvement of both dominant alleles of both the genes. There are two loci, one is A and second is B, and for the both loci, the dominant allele is involved for the this translation of the trait in the form of human skin color. So that capital A, capital B, this will give the negro color. In the case of capital A, capital B, small b, it means at one loci there is a dominant involvement of dominant gene for both, dominant allele for both the genes, and in the homologous chromosome there is involvement of recessive allele for both the loci for both the genes. Then the skin color will be of intermediate type, which is scientifically called as mulatto type. Mulatto type genotype will be capital A, capital B, small b, and this should be B, not A. Capital A, capital B, small b. And in the case of small a, small b, it means both the genes in the case of homozygous and recessive condition, then the skin color will be albino, जो बिल्कुल गोरे होते हैं, बिल्कुल white होते हैं. This is due to only involvement of the all recessive alleles at both the loci. Then it will give the result of albinism or white skin color. We have taken one example to make the crosses between the mulatto colors. If any female with mulatto skin color, जो intermediate skin color ना काली है ना गोरी है, बीच की जो female होगी, if it has been crossed, it is being crossed with the mulatto colored male. अगर ये उसी तरीके के मेल से शादी करती है या क्रॉस बनता है, then after which you may see that this skin color will follow same the phenotypic pattern as it was in the case of multiple factor hypothesis in the case of kernel color in wheat. It means there will be one possibility of one individual where all this dominant allele at both the loci will involve. It means capital A, capital B. This may be of black or negro color. एक ऐसा इंडिविजुअल हो सकता है इस प्रकार के क्रॉस पर जो कि बिल्कुल निग्रो होगा, and there will be chances to produce more individual with dark skin color, and this dark skin color in which the three doses of dominant allele will be involved, including both the loci. Two dominant allele at one loci and one dominant allele and one recessive allele at another locus. Okay, one locus, both dominant allele, another locus, one dominant, one recessive allele. If it is the condition, then the possibility may be capital A, capital B, two will also be possible, and capital A, capital B, small b, two events may be possible. In this way, total possibility of four events will be in the form of dark skin color. And if there is involvement of two dominant and two recessive alleles of both the loci, it means at one loci capital A, small a, so second locus capital B, small b. If it is the condition, then there will be three types of combination. In one combination, capital A, capital B, small e, In second condition, small a capital B should be, it is wrong perhaps over here, it should be small a capital B. Then small a capital B and capital A small b. If this is the condition, then all individuals will be of intermediate type, it means mulatto type. There will be clear cut existence of mulatto type, and this the possibility for this event will be 6. And at last there may be possibility that involvement of only one dominant allele and other 3 alleles including both the loci will be of recessive type. It means at one locus capital A and then small b, or another condition small a capital B small b. If this is the condition, then the phenotype will be light skin colored and the phenotypic frequency will be, it means the possibility of this event will be 4, and only one individual may be there with all recessive alleles small b, and this will be producing white or albino type of phenotype. This is the example of this quantitative inheritance in the case of skin color in human. Now this is the very classical and very much strong example of height in man, which is being governed by the quantitative traits. As in the previous example we have taken, only there was the involvement of 2 pairs of genes for resulting the phenotype, whereas in the case of human height of man human, there is involvement of 10 pairs of genes. It means a huge variability of intermediates among the individuals will be existing, while in the case of this human height, tallness is governed by all recessive genes. As in the case of skin color, the albinism was governed by the recessive genes, and dwarfness is governed by the existence of by all dominant genes, whereas in the case of skin color there will be involvement of all dominant gene for negro color. And here in the same way, tallness is governed by recessive genes and the dwarfness is governed by dominant genes, and they show normal distribution. I have told you that in the case of normal distribution there will be 2 clear cut extreme values, one extreme value before the middle value or the maximum peak, and the another will be the at end point of the maximum peak. With this example you can see the 0 is the maximum peak and the before value this is the first parent, and in the point of this graph, this normal distribution is the presence of the male parent or second parent. These are now I would like to give some points which make clarity among the quantitative and qualitative genes. What are the different points or different clear-cut differences among qualitative and quantitative genes? In the case of qualitative genes, these are governed by trait of kind. Various kind are there. Suppose red color and white color, okay, or round shape and wrinkle shaped. These are the governed by the example of kind. In the case of quantitative genes, these are governed by traits of degree. Various degrees are there. In the case of quantitative traits, they show discrete phenotypic classes and display discontinuous variation. And the quantitative genetic, in the case of quantitative genetics, they display continuous variation. This is the major thing which has to be measured, which required various statistical tool to study the quantitative genes, because they show continuous continuous variation in the case of quantitative genes. And the third one, 2 or more alleles of a single gene. They are governed by 2 or more alleles of a single gene. There is involvement of single gene, but various alleles may be there, providing the different phenotype in the progeny. Whereas in the case of quantitative genes, there is involvement of many non-allelic genes. There is involvement of many genes which are non-allelic. If they are non-allelic, it means they will be the genes, and the crossing over will take place. And now fourth one, least or not influenced by environment. There is very little, or you can say negligible involvement of environmental factor effect in the case of qualitative genes. In the case of quantitative genes, these are environmental effect. These polygenes affect variously. Polygenes are affected variously due to involvement of various environmental factors. And fifth one, constant and ratios are significant tool of analysis. Simply count You can count or simply ratios, different ratios which has been depicted by Mendel. They will be enough to study the quantitative genes, whereas in the case of the qualitative genes, whereas in case of quantitative genes, various statistical method will be. What are the statistical methods which are significant in the case of quantitative genes? I will be discussing in future slides, in the future lectures, in my future lectures. Now, individual mating and progeny test is required for improvement. For improvement of the genes which are governed by the monogenes, the mating and progeny test is enough to make the improvement processes regarding monogenic traits. And the traits which are, most of the traits which are being governed by quantitative genes, there must be there is a necessity to study whole population. Therefore, population with all possible mating, I mean random mating, is useful in the case of this studying the improvement of the traits which are being governed by quantitative these genes or quantitative inheritance. Friends, I have tried to give your glimpses or very clear-cut idea about differences of the qualitative and quantitative traits, and in the public domain, in the case of animal and plant both, most of the traits are governed by various quantitative traits. Therefore, it is necessary to make very clear-cut idea and very clear-cut understanding about the inheritance of qualitative traits. If I'm sure that you have learned something with my lecture, so that kindly like it, share it, comment all the needy student, and if yet you have not subscribed my channel, then kindly subscribe it so that you may become notified with my coming lecture.